Associate Professor Evren Azeloğlu, and esteemed participants. A warm welcome to BioInfo Congress 7. We are truly honored to have Associate Professor Evren Azeloğlu with us today, presenting on multiomics to nanotechnology, how bioengineering redefines precision medicine, an area of profound importance and intrigue. Following the presentation, we'll have an engaging Q&A session where we'll address both questions submitted by our participants and those thoughtfully prepared by our team. We look forward to everyone's participation to enrich the dialogue even further. Dr. Azeloğlu, welcome once again.
Merhabalar, hoş bulduk. Duyabiliyor musunuz?
Evet hocam, sizi duyabiliyoruz.
Merhabalar, teşekkürler. İngilizce-Türkçe karışık bir şekilde devam edeceğim. Konuşmayı muhtemelen tamamen İngilizce yaparım ama soruları Türkçe alabiliriz. Hiç sorun değil.
Dilerseniz başlayabilirsiniz hocam.
Tabii. Eğer bir saniye. Eğer herhangi bir noktada tekrar etmemi isterseniz Türkçe'de de tekrar edebilirim. Teşekkür ediyorum tekrar davetiniz için. Bugün genel olarak benim laboratuvarın çalıştığı alanı iki parçadan tanımlamaya çalışacağım. O yüzden konuşmaya başlamadan önce şimdi İngilizceye geçeceğim ama laboratuvarın yaptıklarını anlatmak istiyorum biraz. So this is our lab. We're about 10 years old, and we sort of have two main foci, two different areas that we focus. One is more systems mechanics, mechanobiology, trying to understand the fundamentals of biology and biophysics using engineering and systems engineering methods. And the other one is system therapeutics. I'm in a department of medicine, so in a hospital. So our goal is to really develop new therapies and new devices. So today I'm going to tell you guys essentially two stories, one from more our fundamental side and how that actually builds up the knowledge of the second topic and how we can build next generation therapies. So before I start, these are my disclosures. I'm employed by Mount Sinai School of Medicine, and these are not quite relevant except for our patent is owned by both Mount Sinai and Columbia University. Okay, so before I tell you guys a little bit about engineering and mechanobiology and how this changes modern medicine, I want to introduce you to kidney disease. This is something that most people are unaware of, so I want to just give you guys a little bit of foundation of why we study what we study. So acute kidney injury is essentially what happens when folks are hospitalized. Unbeknownst to most people, 1 out of 5 adults and about 1 out of 3 children that are hospitalized, that are in a hospital, will experience some form of kidney injury. This is about 15 million people a year. And this has sort of grades. There are more mild injuries. Sort of this purple line that quickly resolves within about a week. And then more severe injuries, as you can see, lead to loss of kidney function over extended period of time. In fact, about 20 to 50% of these AKI patients, acute kidney injury patients, will develop progressive chronic kidney disease or CKD. And this is actually sort of a big deal because about 1 out of 7 people in the US are impacted by this. It's generally a global statistic. And in America, which is very famous for not really caring about patients in terms of healthcare spending, when you have kidney disease, you do actually, the government does take care of kidney disease patients, and it costs the United States government $130 billion a year. We really don't know why people who have injury or certain people who have injury become kidney disease patients later in life, which essentially leads to dialysis and death. and then others really recover right away. So these mechanisms are not known. So we wanted to study this, to understand essentially these 15 million people every year getting this. How come about half of those develop kidney disease and the other half does not? So to study this, we chose a very promiscuous model, a model that a few years ago was really, really widely available, and that was COVID-19 associated kidney injury. So the coronavirus really impacts the kidney, and about 50% of the patients that are hospitalized actually will have some kind of kidney injury. We really don't understand why, but if a patient does have kidney injury, they likely will not survive. Whereas if they do not have kidney injury, they're much more likely to survive. And there are a lot of theories as to why the kidneys are damaged, why they're even infected, because this is a respiratory virus. Why would the kidneys even be damaged? We don't really understand fully, but it does cause this damage, and we had set aside as to with this project. This was early on, a few years ago. We had started essentially early on in the pandemic, right away, to try to understand why some patients develop kidney injury and later on kidney disease and others do not. So we used samples we collected from patients, in this case, COVID patients, but this was not limited. So in two separate hospitals, Mount Sinai in New York and Michigan in University of Michigan Hospital in Ann Arbor. We recruited about 170 patients, mostly with COVID, but also some non-COVID kidney injury patients. Again, some plasma samples were collected, and the goal was to use machine learning and other methods to try to understand what leads to the damage processes. We used a very specific type of proteomics that's called discovery proteomics, TMT16 discovery proteomics. This is an isobaric type of proteomic method that really quantifies the levels of the protein very precisely. It's very reproducible. And we had to invent a new method of quantifying this because we had a lot of patients. So we came up with this pooled urine correlation method where we took 400 earlier subjects and we created loading controls. I'm not going to go into the details, but at the end we were able to get about 6,000 proteins, and we processed 13 patients at a time. So obviously the first question we asked, this was early on in the pandemic that we were collecting these samples. The first question we asked was, can we actually use these samples that we're collecting with the goal of understanding kidney disease? Can we actually use this for other goals, such as, for example, prediction of outcomes? So does the urine that you collect at the time of hospitalization have any value to the outcomes prediction that occur about a week or 2 weeks after the collection of the urine? So for that, we took our patient cohort and we actually divided them into outcomes in a binary manner. We said, do they have kidney injury? If it's yes, then we put them in the severe column. Were they placed in intensive care unit, mechanical ventilation, or did they have to be hospitalized over 3 weeks, essentially a month or more, or did they die? So if we said yes to any of these, we said these patients were a severe case. And if they said no, then we put them as a mild case. And then we split the patients into by a 2/3 level discovery and validation groups. First, we looked to see if the different hospitals had different effects. So the Michigan folks here you can see are spread across the New York folks. So we didn't really see a hospital effect. So that was good. That means the patient characteristics and the sample processing was similar for both cases. Then we said, do we actually see any effect? If the patients have severe outcomes, do they look different than those that have mild outcomes? It was somewhat lukewarm. You could see in this dimensionality reduction space using the UMAP method that they sort of cluster, but far away, definitely away from the healthy folks, but not really separating strongly. But we can actually do differential proteomics analysis from this. We used simple differential protein analytics to look between the severe and the mild outcomes. They actually clustered quite well, and you can see here the severe outcomes to the right, and the mild outcomes are here to the left. This is unsupervised clustering, so we don't actually partition them by hand. This is just mathematically they're partitioned. And you can see that most of the impact is to the right. And from that, we can actually identify which proteins are in the urine correlate with severe or mild disease. So we obviously wanted to ask what these proteins do, where they come from. This was pretty important. So we looked at these proteins separately, downregulated versus upregulated. The downregulated guys, interestingly, were very much related to cell adhesion. So, and this was surprising because this was more of the latter points collected by other groups that have looked at proteins in the plasma, particularly for severe COVID injury, and the upregulated proteins were also related to adhesion, but indirectly. And here you can see extracellular matrix disassembly. So the cells are essentially wanted to detach. And if you look at which cells the signals come from, you can see that podocytes, these are specialized kidney epithelial cells that I'm going to talk to you about later. My lab studies these intently. Most of our projects are focusing on these. They actually rank number one. So the cells, podocytes, and you can see your proximal tubules, mesangial cells, and endothelial cells. These are cells of the kidney that were lighting up. So we wanted to run a classic, simple machine learning algorithm. So we use random forest with tenfold cross validation to see if we can predict. This was the discovery set, and the validation set kind of held the AUC appropriately. This is a Boruda selected feature list from the 6,000 proteins that we have identified. We actually only 12 of them had a decent importance, so we used those. And you can see that the patients did really segregate properly to high risk and low risk. And if you do a tertile split, you can actually predict a good outcome by 92% accuracy and a bad outcome by 85% accuracy on individual tertiles. Now, this is prediction. We are really not that interested in prediction, especially anymore, because we don't have a lot of acute COVID injury, even though this is still important. Now we have other methods to stall outcomes in COVID. So we wanted to ask this question that do we have this urinary biomarkers, essentially proteins that we detected in the urine? Can we actually use the same 90% sensitivity cohort for other kidney injury? Now, this is the important part, right? This is the 15 million or so people that I mentioned that are injured every year in hospitals and later go on to develop kidney disease. So can we actually predict those guys? And for that, we went back to our own cohort. This is the severe COVID-19 cohort. When we actually put the non-COVID related acute kidney injury, the markers really recapitulated almost entirely, except for these two guys. And these two guys are actually special because they're not lung markers. They're not kidney markers. Excuse me. They come from the lungs. So these are actually respiratory markers. So every single one of the kidney markers recapitulated in the non-COVID AKI setting. In fact, when you project the non-COVID AKI cohort here in blue cyan color, you see that they cluster even better using with the same marker set. And these are classic markers of kidney injury, cystatin C or cystatin 3 or albumin, and you see that they actually don't work as well as our 12 marker proteomic method. We went to another cohort. This is another project that I'm involved with. It's called Kidney Precision Medicine Project, or KPMP. It involves 50 different hospital sites in the United States and several in Europe to get to understand kidney disease. And we went to their biomarker set and looked at the acute kidney injury cases within KPMP. We saw that the markers that we have detected actually almost entirely recapitulated in the KPMP cohort as well. This was very encouraging, but we do have this marker set that is predicting things. Wonderful. and that seemed to be strong correlates. But did they actually mean anything? What do they predict for? When we look at the disease process enrichment, we see that they actually mostly correlate with, again, cell junction assembly and cell adhesion. So this is how cells are adhering to each other in this case. And we see a few other processes showing up here: renal filtration, cell differentiation associated with that, and glomerular epithelial cell differentiation, which is essentially a fancy way of saying podocyte differentiation, as I have noted before. So these seem to be, there seems to be a signal coming strongly from podocytes. So we actually went back to the KPMP data set and asked if these markers are coming from any individual cell type. And we can do that because KPMP has performed biopsies. So they've actually taken kidney tissue out of the participants and performed single cell RNA-seq. And we have actually published with this data recently. We had Cell Advances and Science Advances and a Nature paper that came out a few years ago on this. So we went back to that database and asked the question, do these biomarkers come from specific kidney cells? So we looked at the specificity signal here. And what we saw was they were mostly coming from glomerular cells, right? So these are the glomerular endothelial cells, podocytes, and parietal epithelial cells. These three cell types are kidney cells that are specifically strongly expressing these markers. So there's definitely glomerular injury. There's definitely kidney injury that is more on the glomerular axis, which we think is more sensitive to cell adhesion related processes. And then there's here are the rest of the nephron segments. This is essentially the rest of the kidney. So now this is with only the 12 markers. These are predictive, very good. They come from the urine, so they are potentially powerful. They can be used in the hospital setting, but not necessarily the whole picture. So we said, why don't we take this method now apply to the entire urine set and the entire plasma protein set that we have obtained. And this is a lot bigger patient cohort. Now we're looking at about 1,000 patients total, looking at both plasma and urine. And I color coded on the left here, sort of this pink red hue is the plasma and the green color is the urine. And you can see here, as you move across the nephron, so this is essentially how the cell filter, the kidney filters and forms urine. First, it's in the blood, so you have endothelial cells. This is the blood vessels. And then it goes into the glomerulus, the podocyte, the parietal epithelial cell. And then it's created into a filtrate, runs through the rest of the nephron, and gets filtered out. So you can see, first of all, the urine markers are highly overrepresented in the glomerular segment, and the blood markers are highly overrepresented in the interstitial and the immune markers. So if you're studying an immune process, the plasma tends to work better. And if you're studying a cell core organ process, then urine sample seems to be working much better, which actually explains why people who have worked with plasma proteins before were unable to create mechanistic, reproducible mechanistic understanding of gene markers that are associated with tissue damage. So we used dynamic enrichment, which is a process that we've come up with some years ago and used with KPMP as well, to see if these differentially expressed proteins in the urine and the plasma actually work together in cell biological processes. And this uses the MBCO, molecular biology of the cell ontology. So it's an ontological system. And what we noticed was it's really coherently focusing on cell communication, extracellular matrix homeostasis, cell adhesion. And then here you can see towards the left, more so on blood-related processes, coagulation, fibrinolysis, and complement system. Okay, so this core was really interesting to us because, again, just like our predictive markers, cell attachment, cell adhesion, and cell-cell communication was coming out as the top process. So here I'm just showing you where it's sort of in a linear manner, all these different processes. And you can see here adhesion, cell adhesion, regulation of cell adhesion, all the same processes coming out, and very strongly in both urine and plasma. So now to really understand the deep sort of tissue level process, what you need is tissue biopsy, right? So you need to stab the organ, in this case, the kidney, and take some tissue out and try to understand if these are true. Now, obviously, we can't do that, at least for thousands of patients randomly, because there are risks, stabbing somebody's kidney. But we actually have a tool that pretty much comes close to stabbing someone's kidney, and that is the cells that are already in the urine. Fun fact, when you pee, you actually pee out some of your cells every time. So what we did was we took the urine, we precipitated and collected all the individual cells within the urine and run single cell RNA-seq on these urine sediment cells. And we now can identify where these cells come from. Most of them are obviously blood cells, PBMCs, monocytes, mononuclear cells, T cells, and so on. But then a lot of kidney cells are also shedded, and we can actually look at those individual kidney cells and see which processes are highlighted and which ones. Then we can go back to our network analysis, and this time include the single cell RNA-seq as well, and create an interaction score between individual cells, the networks that we obtain from individual cells, the genes, and the genes we obtain from the plasma and the urine. And when we use this, we noticed that the loop of Henle was strongly implicated, and that this is the part that is one of the most easily damaged parts of the kidneys. So it was actually sort of reassuring that we were identifying sort of these loop of Henle cells. And if you actually look at the processes that are involved for these highly enriched genes, you see that cell adhesion via plasma membrane adhesion molecules comes number one again. And here's the network that comes out. And I wanted to specifically highlight here these particular five genes. These are the 5 of the 12 that we have originally identified as the predictive genes for our AKI model. And cystatin C is here. It's already being used clinically as a predictive marker. So as many of you are aware, if you try to publish something like this, you're making big, grand claims. They say, well, you have to validate this somehow. Unfortunately, you really cannot validate this unless you really have to take samples out of a person. So we wanted to come as close to that as possible. So we created kidney organoids from the blood cells of these patients. So here are our kidney organoids. And those of you guys are mostly familiar with this. There were several talks, even in this by Congress, about these organoids. These are essentially early differentiated systems that are three-dimensional, that recapitulate most parts of the kidney. Here, just I'm showing the glomeruli and the tubules, the main segments of the kidney, lighting up with their different markers. These are the little LOM segments. These are the little tubular segments. And here you can see it in 3D where these structures are. So we have these kidney organoids from the patients. Then we wanted to ask, can we actually infect these organoids, and can we actually? And the answer was already known. This was published also during the pandemic, that people try to understand this. So they made lung organoids and kidney organoids, and they actually are infectable. And we actually can. And here you can see we have 2 patients here, and one of them was not infectable, and the other one was infectable. So we actually used this one as a model. Then we ran single cell RNA-seq. To see where these segments are, which cell types are infected and where they were. The C1-C10 segment was infected. These are the interstitial cells, and the podocytes and the tubules, the master kind of functional workhorse of the kidney, and they were also infected as well. And when you do immunofluorescence, you can actually confirm this. This is the ACE2 receptor that is the main target of the virus to insert into the cells. This is the nuclear protein, nuclear capsid protein of the virus. So you can actually see co-localizing with LTL, with the tubular markers, and you can see that the tubules being infected locally within the kidneys. So now we have a model system we've established. We can see the infection. Can we actually understand what's happening? Can we recapitulate the processes? So for that, we took the tubular cluster, we analyzed that individually in the single cell RNA-seq, and we took the podocyte cluster, we individually analyzed that. And here you can see focal adhesion and cell adhesion processes again coming on top. Then we took these differentially expressed genes and inserted into the network that we have identified using protein and urine and protein, plasma proteins that we've identified. And lo and behold, they actually go straight into the same processes that we identified with urine and plasma. And you can see here extracellular matrix homeostasis ranking number 1, right in the center, and cell adhesion here in the periphery, and cell communication, intracellular degradation here connecting through collagen biosynthesis and degradation. So the network holds, and if you enrich for it, extracellular matrix organization, extracellular structure organization, again, the top process. But not only this, we can actually then now pull receptor ligand interaction and tell you which specific matrix proteins are identified and which are specifically targeted in AKI. So based on this, we think that we have identified the master sort of pathway that is dysregulated during kidney injury, the acute kidney injury clinically, that can potentially be not just predictive of which patients are potentially going to develop chronic kidney disease, but also potentially targeted after kidney injury or during kidney injury to protect the kidneys for future degradation. Now, this I want to just closely recap some of these topics that I covered in the past few minutes. I showed you guys that you can take liquid biopsy samples, plasma, and urine, and you can use machine learning to actually not just predict kidney outcomes in severe acute kidney injury, but also build a database of really large database of unique proteins that can give us more additional information for study. This is freely available, by the way, to study other processes related to kidney injury. The accuracy of prediction about 1 to 2 weeks prior to an event was about 90%. This was specific for COVID-associated AKI, but we think that they are universally adoptable to AKI, acute kidney injury in general. And our integrated methods actually identify gene networks that we think not only are impacted by COVID infection, but in general, severe kidney injury that can be targeted and used for prediction or treatment of kidney disease development in the future. So with this, since I know your Congress does really broad interest cover all biology, I wanted to turn around at this point and sort of in the midpoint and talk about this other aspect of our lab that we study, something almost completely different, and talk about some cancer metastases and how we can study that using mechanobiology and systems engineering methods. So for that, I actually want to remain in the kidney for 2 seconds because it shares the story of our origin story. We published a few years ago this Nature Communications paper where we showed we can use microfabrication. This is essentially performed in a clean room and create these robust kidney glomerular differentiation surfaces. So this was published a few years ago. And this is just to sort of show you guys how we can change cells in culture from their usual phenotype into this sort of arborized phenotype that really closely mimics cells in the body. And we've shown that this differentiation process is super rapid. Usually this differentiation takes about 2 weeks in culture, that we can perform it within 2 days almost. At 4 days, we have rapid differentiation with appropriate cytoskeletal markers. And we have also shown that these differentiated cells are actually a lot superior to regular on-pattern differentiation cell, and this data is unpublished, where the paper is in revisions right now. But just to show you guys the origins of this project, I wanted to show you what we've done with kidney cells and how we came up with this idea. So I had a very, very talented graduate student who was working with the system, who came up with the system. And during one of our conversations with her, I asked her, um, I'm wondering if we can take this system and use it for uh, studying other cell types. Particularly, what was interesting with our system, if you guys remember here, has these sort of fine channels where cells, if you look at podocytes, they form these processes and they stay where they are. In fact, in our paper, we show that they don't cross these channels ever. I was asking my student if she thought maybe if we cultured, let's say, cancer cells, if solid tumor cells would traverse through these. So she actually took it really seriously. So this is a video that I'm going to show you that we obtained that she took at the Howard Hughes Medical Institute Center using a mosaic lattice light sheet system. There's only one of these systems in the world. So Eric Betzig, the Nobel laureate, built it personally, and she used that microscope to capture. And this is an LM24 MJ231 cell line. It's a breast cancer cell line, triple negative, that is on our system. And here I'm just showing you the cell reconstructed with a live actin marker. And here you can see it squeezing through the channel, the microchannels that we have fabricated. Again, remember, in the video that I showed you earlier, kidney podocytes, the kidney epithelial cells that are not tumor cells, they actually don't do this, right? They sit and they spread and they form processes, whereas these triple negative breast cancer cells, they actually are a lot naughty. They squeeze through and penetrate. So this was very interesting, but it turns out biomechanics is important, not just for kidneys, but also for cancer. Kidney, particularly metastatic cells, use what's called an invadopodia structure that helps it penetrate through sheets of extracellular matrix and looks at squeezes its nucleus, which is something that most healthy cells cannot do. And using that, it can actually extravasate and enter blood vessels or exit the tissue matrix that's encapsulating them. So my student, Smriti Bhattacharya, graduated a few years ago. Was a very successful student. She's now the director of NYU Nanofabrication Facility. Was really curious, so she talked to an oncology group, got some non-metastatic cells and some metastatic cells, and ran this experiment. And lo and behold, we did see that the non-metastatic tumor cells, indeed benign tumors essentially, indeed perform just like kidney podocytes, which is the system that we usually use to study, whereas metastatic cells traverse around as if there is no pattern. And you can quantify this quite accurately, and you can build an invasion index, whether the cells invade or not, and normalize it to the amount of motion that the cells exhibit. So this is very important because it actually takes the motility of the cell out of the equation and looks at purely the invasion, which is very important. And you can actually count the number of invasion events for cells, or really do sophisticated single cell analysis with the system. When we looked at this, we were, again, collaborating with an oncology group. They were giving us these cells because they have done other assays. So we said, can you guys help us run these assays so we can actually see what kind of difference you get between invasive and non-invasive cells? And they ran the assay. It's called the Transwell assay. It's essentially a 3-dimensional migration assay. And a counter assay, which is a lot simpler. It's a 2-dimensional assay where you plate the cells, scratch in the middle, and let the cells kind of migrate into the space. So, and they said, here are 2 cells. This is what we do. And we looked at their scores. It's like, okay, cool. You guys can quantify this. We asked, Smriti asked, what score do I get when I do it on our system? And this is the index we get. So when you actually look at the difference, you see here, the difference between the non-invasive cell and an invasive cell in the micropattern assay was about 50-fold. So your quote unquote sensitivity is about 50x. Whereas in the other assays, you're looking at 2 to 10-fold sensitivity. So it was really, really superior to the assays that are existing assays. metastatic invasion assays. So she asked the question: is this across the board, universally applicable to all the cells? She took a whole bunch of different solid tumors and ran this, and indeed, it was. When you look at that, look at the scale of invasion index versus the average absorbance in the transwell. The transwell was more of a continuity. So yes, these sort of less invasive cell lines were showing up less. They were exhibiting less absorbance compared to these invasive cells. But look at the difference. It's like night and day. Less invasive lines here still have a number, whereas in the invasion index of the micropattern assay, they absolutely rank close to zero. So really, the system is able to say like a 0 and 1, almost in a binary fashion, whether cells are invasive or not. So we wanted to follow this. This was actually correlated with their nuclear stiffness, right? Because people assume that that's the primary driver. And we saw that it did correlate, right? Indeed, the cells that were less invasive did have stiffer nuclei, and the more invasive cells did have softer nuclei. But it was not it. It was not explaining fully. So we wanted to change the nuclear stiffness to see if it actually would successfully alter this. So we used this HDAC inhibitor, TSA, which essentially uncoils the DNA and allows the nucleus to be more compressible. And we evaluated that with using atomic force microscope indentations in the nucleus. So we show that we can take a non-invasive cell, this is the MCF7 cell line, and by treating with the TSA, make their nucleus softer, which interestingly slightly increased their decrease their migration, total migration, but increased the invasion index. This was really surprising to us because the two were seemingly contradictory. And at that point, Sveti walked into my office and said, now I understand why this chemical is such a heated, kind of creates this heated debate. And she showed me these really high impact publications claiming the exact opposite. Half of them were saying TSA was reducing invasion, and the other half metastasis, and the other half was saying TSA is increasing metastasis. And we now understand. And we went back to the assays that people used to decide whether it was increasing or reducing it. And indeed, those who were using migration as their criteria, they were noticing a decreased metastatic potential. And those that were looking at three-dimensional invasion were seeing increase. So this was very encouraging. And here, Smithy also wanted to show that you can completely separate the effects of invasiveness and motility in another manner. We're using drugs that are known to impact metastatic potential. And here she shows that here, for example, in the specific case of the IGF-1R inhibitor from Bristol Myers Squibb, that you can reduce both motility and invasion index at the same time. So this paper came out in Science Advances just this year, last year, 2024. So if you're interested, you can follow up on further details. In fact, if you're interested, I just wanted to show you a little glimpse of what we're currently working on. These are NDA-231 cells invading in our micropattern assay, lit up with a specific cholesterol biosensor. And here you can actually see, as these cells are trying to invade, here there's a squeezing cell, how lipids are interacting with different cytoskeleton proteins to actually allow invasion. You can see in different cells how lipid cytoskeletal interaction is playing out. This is what we're studying actively right now. And if any of you are interested in doing some science on this regard, I am looking to recruit a postdoctoral fellow or a graduate student who's interested in working on this project further. So to wrap up, I want to show you on the other side of the spectrum, micropattern surfaces can be used to rapidly quantify metastatic potential. you can actually detect effects of nuclear elasticity, contractileness, motility, and evasiveness separately, and you can actually dissociate the effects of the drugs on these different biophysical attributes successfully, which we think will have very strong clinical use. With that, I want to acknowledge my lab and our collaborators, particularly Sminty here, who's an alumni at NYU right now, who ran most of the work, and a junior faculty member, an assistant professor in the lab, Nandita, who ran the biomarker project along with another PhD student in the lab who helped both of the projects. And I will be happy to take questions.
Dr. Azeloğlu, thank you sincerely for this enlightening presentation. With that in mind, I'll now proceed to the first question from our Q&A session prepared by our team and gathered from our participants. While you have observed that drug-induced pathways are similar in both healthy and heart failure patient-delivered cardiomyocytes, could other factors like aging, comorbidities, or immune response provide alternative explanations for these findings?
Güzel soru. Kardiyomyosit değil ama böbrek hücreleri ama soru güzel bir soru. Evet, istatistiksel olarak bunları normalize ettik tabii. Hem hasta olan hem kontrol hastalarında olabildiğince normalize ettik bunu. Ama tabii sıfırlamak mümkün değil. Mutlaka bunun da bir etkisi olabilir. Güzel bir soru.
Cevabınız için çok teşekkür ederim. Bir sonraki soruya geçiyorum. COVID-19 kaynaklı böbrek hastalarında tübüler hücrelerin yanı sıra juxtamedüler nefron hücrelerinin erken dönemde etkilenmesi viral enfeksiyonun böbrek üzerindeki etkilerine dair alternatif bir açıklama sunabilir mi?
Evet. Evet, ya biz bunun böyle olduğunu düşünüyoruz. Bu alternatif bir açıklama olabilir. Bence yani bizim datanın gösterdiği hem bütün hücrelerin bağlantılarını etkiliyor. Ancak özel olarak juxtamedüler hücrelerde güzel bir gözlem bu. Bu hücrelerin daha kolay etkilendiği bilinen bir şey. Örneğin böbrek rahatsızlığında, akut böbrek rahatsızlığında oksijen seviyesinden ilk önce kaybedilen oksijen seviyesinden ilk etkilenen hücreler bunlar. Potansiyel olarak o yüzden bu hücreleri görüyor olabiliriz. O yüzden hücre bağlantısı, hücre ve hücre matrix bağlantısını ilk önce kaybeden hücrelerin bu olmasının nedeni o olabilir. Güzel bir gösterge.
Cevabınız için çok teşekkür ederiz. Bir sonraki sorumuza geçiyoruz. Bu sorumuz YouTube'daki katılımcılarımızdan geliyor. 130 COVID-19 örneğinden 6'sında tespit edilmesi göz önünde alındığında COVID-19 ile ilişkili böbrek hasarının doğrudan viral enfeksiyondan ziyade sistemik inflamasyon veya immün yanıt gibi dolaylı mekanizmalarla daha fazla ilişkili olabileceği konusunda ne düşünüyorsunuz?
Evet. Bu kime sorduğunuza bağlı. Almanya'da çok büyük bir grup var. Onların başını çektiği bir grup. Direkt olarak kimi hastalarda bunun olduğunu söylüyor. Almanya'nın genelinde topladıkları bir sürü bir raporu çıkarmışlardı. New England Journal'da çıkmıştı yanlış hatırlamıyorsam. Daha sonra Lancet'ta bastılar bunu. Sürekli ısrar ettiler yani böbreği direkt. Ama biraz azınlıktalar. Harvard ve Columbia'daki grubun iki grubun kağıdında biz 1000 tane hastanın böbreğine baktık. Hiçbirisinde bulamadıkları dediler. Biz işin o kısmına girmedik. Yani immün bazlı mı en direkt bir şekilde yoksa direkt olarak böbreğe gelen bir virüs yüzünden mi bu hasar mekanizması çalışıyor? Bunu cevaplayan bir deney yapmadık. Ama daha pragmatik yaklaştık buna. Hangi vasıtayla olursa olsun işin sonunda hücrelerin birbirine bağlanmasının engelleyen bir sinyal ortaya çıkıyor. Ve bu yüzden böbrek hücrelerinin yavaş yavaş birbirinden çözüldüğünü görüyoruz. Bu eğer idrardaki toplanan hücrelere bakarsanız bunun zaten genel olarak hem Almanya grubunun hem Amerikan gruplarının gördüğü hücrelerin çözülüp daha fazla idrar yoluyla atıldığını iki taraf da kanıtlamış durumda. Yani bu bilinen bir şey zaten. Biz yalnızca bunun hangi genetik hangi proteinler üzerinden olduğunu gösterdiğimizi iddia ediyoruz. Ve eğer ki haklıysak bu genel olarak evrensel olarak bütün hücrelerin hepsine bütün böbrek zararına hastalıklarına atıf edilebilir. İşte o zaman zaten bu moleküllere karşı bir ilaç geliştirilebilir diyoruz.
Evet, cevabınız için çok teşekkür ederiz. Bir sonraki sorumuza geçiyoruz. Bu sorumuz YouTube'daki katılımcılarımızdan geliyor. How do you foresee the future of personalized medicine evolving, particularly with the advancement of nanotechnology and the integration of diverse omics data?
Çok güzel, çok güzel bir soru gerçekten. Gelecek çok parlak eğer dünya kendi kendisini yakıp bitirmezse mutlaka bunların personalized medicine, precision medicine, kişisel olmasa bile gruplara dair precision medicine'ın gerçekten bir hayal olmadığını düşünüyorum. Önümüzdeki 10 sene içerisinde gerçek olacak bence. Yani hastaneye geldiğinde direkt hangi gruba dair olduğunu, hangi ilaçlar alması gerektiğini bileceğiz.
Anladım. Çok teşekkür ederim cevabınız için. Bir sonraki sorumuza geçiyoruz. Bu sorumuz da YouTube'daki katılımcımızdan geliyor. Are there nanotechnology techniques that could potentially move from molecular level discoveries to real clinical impact for patients? Or what kind of approaches is needed? Thanks.
Çok güzel soru tekrar. Birkaç sene önce hemen pandemi öncesinde basmıştık bunu. Nature Reviews'ta bir makalemiz vardı. Bütün bu teknolojileri iki eksen üzerinde görselledik. Bir tanesi ne kadar komplike, ne kadar gerçek fizyolojiyi yansıttığını gösteren bir eksen. Örneğin bu organoidler gerçekten komplike, her şey birçok fizyolojik gerçeği ölçmenize yardımcı oluyor. Ama öbür eksene esnekliğini koyduk. Yani ne kadar çok yapabilirsiniz, ne kadar ucuz, ne kadar kolay karşılaştırabilirsiniz. Matematiksel olarak ne kadar istatistiksel olarak ne kadar büyük gücünüz var. Bunu koyduk. Eğer bu şekilde düşünürseniz organoidler böyle sol üst köşede, evet çok komplike ama yani mesela bizim normal bir hücre kültürü yapmak için kullandığımız media $20. Organoidler için kullandığımız media $750. Yani bundan bir tane yapıyoruz, öbüründen 10.000 tane yapıyoruz. Doğal olarak hücreleri kullanmak daha kolay. O yüzden hücreler sağ alt köşede duruyor. Hiç komplike değiller. Aldığınız sonuçların %99'u çalışmıyor insanda. Burada da organoidler var. İnsanda çalışıyor ama 10.000 tane yapamıyorsunuz. Bir tane yapıyorsunuz. Amaç teknolojilerin bu şekilde hem esnek, ucuz hem de buna cevap verecek geliştirdiğiniz ilaçları görebileceğiniz bir yere geçmesi gerekiyor. 2 örnek verdim. Bence bu 2 örneğin birleştiği gün gerçekten Bu denediğimiz ilaçlar candidate olmak yerine gerçekten hemen bir adımda insana gelecek.
Cevabınız için çok teşekkürler. Bir sonraki sorumuza geçiyoruz. Bir sonraki sorumuz da YouTube'daki katılımcılarımızdan geliyor. Given the difficulty of monitoring pathology development in patient samples, does it make sense to model the AKI-CKD process with nephron organoids, or are there insurmountable limitations?
Soru. Ekonomi. Şu anda. Önümüzdeki insurmountable, aşılamayacak tek limit ekonomi bence. Organoidler tabii biraz daha böyle fetal, daha az gelişmiş böbrek hücresi. Onun da bir sorunu var. Daha yetişkin yapıyı yansıtmıyorlar %100. O yüzden organoidde çalışan bütün ilaç adaylarının insanda çalışacağını garanti edemiyoruz ama sorun şu, %90'ı bile garanti olsa, %90 değil, muhtemelen %50 gibi bir rakamdayızdır tahmin etmem gerekse. Çok pahalı olduğu için yüzlerce ilaç adayı deneyemiyoruz. Onlarca deneriz belki. Onlara indirmek için de 10 binleri deneyebileceğimiz bir sisteme ihtiyacımız var.
Anladım. Cevabınız için çok teşekkürler. Bir sonraki soruya geçiyoruz. Bu sorumuz da aynı şekilde YouTube'daki katılımcılarımızdan geliyor. In your COVID-19 study, what was the impact of comorbidities or severity outcomes in the sample population? For example, any unexpected mutations increasing severity.
Evet, ve klinik tecrübesi olan bir izleyicimiz bu. Güzel soru. Komorbiditilere baktık iki grupta. Hiçbir tanesi istatistiksel olarak öne çıkmadı. Tabii bilinenlerdeki sinyal oradaydı. Mesela erkeklerde bilinen bir şey daha zayıf erkeklerin böbreği. Erkeklerin birçok şey daha zayıf ama böbrekleri özellikle kadın böbreğine göre daha zayıf. Mesela bu sinyal olarak vardı. Bunların elimizden geldiğince normalize ettik. Bu istatistiksel gösterdiğim rakamlarda bu denklem vardı yani. Bu yapay zeka sisteminde bunları ayrıca tek tek denedik. %50'nin biraz üzerinde çıkabiliyorlar. %60'a falan getirebiliyor mesela. Yapay zeka ile bu diğer veriyi eklediğiniz zaman %60 gibi bir rakam geliştiriyor ama bu biomarker ile yaptığınız zaman %90'a çıkarabiliyorsunuz bunu.
Anladım. Cevabınız için çok teşekkür ederim. Bir sonraki sorumuza geçiyoruz. In your COVID-19 study, were there any challenges you encountered with your ML model, like imbalances or over-bias in the datasets? What other methods did you use to validate the results?
Çok güzel soru. Teşekkür ederim. Yapay zeka uzmanı bir arkadaş soruyor bunu herhalde. Evet, bu hem imbalance hem overbias sorundu. Bunu ikisini de çözmek için bayağı bir uğraşmamız gerekti. Validation için 10x cross validation haricinde bir grubu virgin bıraktık. Hiç dokunmadık onlara. Kesinlikle analize girmedi. En sonunda onların üzerinde test ettik. Ve o testte %10'a yakın bir kayıp vardı. Başarı kaybı. O yüzden overfit olduğunu düşünmüyoruz modelin.
Anladım. Ve bu son sorumuzdu. Çok teşekkür ediyoruz. Dr. Azeloğlu, thank you very much for taking the time to share your knowledge and for your informative presentation on behalf of myself, our team, and all the participants.
Ben teşekkür ediyorum tekrardan davetiniz için.
We wish you all the best for your health and success in your studies.